Yes, sir.
So very good morning to my class. I am Professor Rambir Singh from Mizoram Central University. So Samara, you are the only candidate in the class?
Yes, sir. There is a one candidate.
Okay, good.
She's from America, so right now she's not joining with us.
Okay, very good. So what is your master degree program? Like what master degree did you did?
Sir, I have done a master's in zoology.
Masters in zoology, very good. And now you are doing PhD in biochemistry.
Yes, sir.
Okay, very good. So we have to discuss some part of instrumentation. Have you gone
Yeah.
through the syllabus?
Yes, sir. I have the syllabus with me.
Okay. So can you just share it if I just because I to just look for it and we can start discussing.
Okay,
You are
sir.
on
Okay, sir.
computer or you are on the laptop or you are on the mobile?
Sir, I'm on the mobile. Right now I'm in my college.
Okay. But that's okay. But I think it will be better if we attend classes on computer because you will be able to understand it more.
Okay, sir.
You have your syllabus ready with you. Can you share it?
Yes, sir. I'll be sharing it with you
Just
in
do
a
it.
while.
Mm-hmm. Yes, anyhow, no need to share it now. I have taken it out. Huh?
Okay, sir.
Okay, I have
Sir,
taken it
I've
out.
shared it on the WhatsApp group.
Okay. So I just share it with you. You can just see the portion which we have to cover up. Now, can you see that?
Yes, sir.
Okay. So largely we have to discuss about chromatography, then part of electrophoresis. Okay.
Okay, sir.
So I hope since you are a student of zoology, you might have heard a little bit about instrumentation.
Yes, sir.
So you know what is chromatography?
Yes, sir.
What is that? Just explain.
So we use chromatography to separate the components in certain solutions or
Now, what is the application part of it?
application part of it, sir. It is used while detecting certain solutions in some samples.
Okay. Now, what is its application in biochemistry?
Hello? Yes, sir.
Yeah, what is its
Can you please
application in
repeat?
biochemistry?
Sir, it's application. Sir, I have done some certain experiments on HPLC in my college.
Okay, very good.
Okay, sir. And sir, and that we detected vitamin D samples from the
Vitamin D.
blood.
Okay, very good. So I hope you know that the word actually the chromatography came from a Greek word, and I just share one presentation with you, and you just go through it. I will share the presentation. Can you see it now?
Yes, sir.
Okay, and you just I'll share this presentation with you. You just go through it. I think now it is on. Now it is on full screen mode, so you can see it clearly, I think. Huh?
Yes, sir.
So it is principles and applications of chromatography. And the word chromatography, it is derived from a Greek word. Chroma means color and graphene means to write. So it's basically a laboratory technique of separation of mixtures. So possibly you know that if you want to do some kind of analysis, then first the molecules must be separated. Because unless and until we have isolated 100% pure molecule, the molecules cannot be characterized. You know what are the techniques of characterization and what are the technique of separation. So you must be able to understand when we talk about the techniques of separation, we have to separate or and purify the mixture, individual component from a mixture. So you know what are the three major techniques of purification? Yes.
So like
Not
sedimentation, evaporation, like this.
sedimentation is I can say like we got another technique which is derived from sedimentation. You can say that is centrifugation, right?
Yeah, yes, sir.
So okay, so these are three techniques basically. One is centrifugation, another is chromatography, and the third one is electrophoresis. So these are the
Okay, sir.
separation techniques. Okay.
Okay,
And,
sir.
and
Okay, sir.
the job of this technique is that we have to purify a molecule in single purified form, and that molecule can at later stage it may be characterized, right? So when we talk about biochemistry, that we know that you know that there is a concept of biomolecules. I hope you are very much aware of that.
Yes, sir.
So what are the, you have primary biomolecules and secondary biomolecules. Sometimes we give a word called primary metabolites and secondary metabolites. You have that
Yes, sir.
idea? So what are the primary
Yes,
biomolecules
sir.
we have in living system?
So there are proteins, carbohydrates, fats.
Yes, yes. And one more. We
Protein.
have four primary biomolecules: carbohydrates, proteins, fats or lipids, and the fourth fundamental molecule is it is
Sir, I
nucleic
don't know.
acid. It is nucleic acid.
Oh, yes, sir.
Okay.
Yeah, yeah. Yes, sir.
So these are primary molecules. We call them primary because these are essential for the survival of the cell. So if you take out
Yes, sir.
anything out of the cell will die and the living system cannot sustain. Living cell cannot sustain. Then we have secondary metabolites. So secondary metabolites are, can you define secondary metabolites?
So secondary metabolites are the things that are produced by the primary ones.
Oh, okay. Any other answer? Actually, these are derived molecules and these are
Yes,
not
sir.
essential for survival. They give survival
Yes, sir.
advantage to the organism, but these are not essential for survival. Okay. So when we
Okay, sir.
talk about biomolecular analysis, whether primary biomolecule or secondary biomolecules, generally for any kind of say disease characterization, we need to identify the molecule which is responsible for the development of disease. Okay. So for
Okay, sir.
identification of that molecule which is responsible for development of disease, we must be able to characterize it. Characterize means we must be able to identify its structure. Like characterization is of two types. One is say concentration based measurement and another is structure based measurement. Okay. So when we talk about
Okay, sir.
concentration based measurement, we know that concentration of a biomolecule may be responsible for a disease, isn't it?
Yes,
Like,
sir.
can you give any example where the disease is associated with the concentration of a biomolecule, change in concentration of a biomolecule? Can you name any disease?
So like certain diseases occurs, for example, the cancers,
Mm-hmm.
with
So
less or more expression of
how
certain
come you
genes.
can link cancer with a biomolecule concentration?
So we detect the concentration of, for example, we check the gene expression, that whether
Mm-hmm.
it is expressing or not, or if it is expressing, then in what amount it is expressing and producing the certain proteins.
Okay, so Smarth, I was expecting some very simple answer from you because like biomolecular concentration and establishment of cancer is still challenging. But yes, definitely there are reports. But since you will be doing maybe a lot of work in clinical diagnostics in your PhD, I do not know what you will be selecting for your PhD. But yes, I'll give you a very simple example. Like you talk about the concentration of glucose. Now, can you link the
Hmm.
concentration of glucose with diabetes?
Yes, sir.
So how? How you can link the
Sir,
concentration of glucose with the diabetes?
in diabetic patients, there's the excretion of glucose when there's when the glucose concentration in the body rises, it just gets excreted out in the urine. So we detect the concentration of glucose there in the urine and decides whether the person is diabetic or not.
Okay, but do you have any assay where we are doing the urinary is it a foolproof assay? You know what is the most common assay for diagnosis of diabetes? And certainly it is not from urine.
Sir, I don't know. I don't remember right now.
Like you must have read, no? Like we go for either fasting blood glucose measurement, or we got random blood sugar measurement, or we got PPG assessment, post-parenteral glucose
Yes, sir.
assessment, isn't it? So these are the
Yes, sir.
commonest assay by which we assess the diabetic condition. Okay.
Oh, yes,
So
sir. Yes,
like
sir.
this is one kind of disease, one kind of measurement where the disease can be established by quantitative measurement of a biomolecule. Now, how it is possible, like how it was established that glucose is responsible for diabetes, elevated concentration of glucose is responsible for diabetes. It is by years of research. Okay. So in these
Okay,
kind
sir.
of technique, we only need to go for quantitative measurement of a biomolecule. There is no need for isolation of a biomolecule, right?
Yes, sir.
But you talked about cancer because glucose is a well-established biomarker for diabetes, right?
Yes,
If
sir.
you want to establish a biomolecule for a particular disease, first this biomolecule need to be measured and need to be identified properly. And for sorry, I'm having little bit of cough actually. Just a second. Huh? Okay, huh? Can you hear me?
Yes, sir.
So for establishment of a marker compound, first we need to isolate that marker compound and then we characterize that marker compound. Now suppose if for cancer, if you want to establish a marker compound, now can you name any marker compound which is established for cancer, diagnosis of cancer? Some proteins have,
Sir,
did I just give you
alpha
a,
fetoprotein, I've heard.
which one?
Fetoprotein, alpha fetoprotein.
Okay. I give you a very common example. You must have heard about a protein called P53 protein, right?
So can you please repeat?
It is P53 protein, P53, protein
Yes,
53. It's
sir.
one of the most
Yes, sir. The
common
tumor suppressor
tumor
one. Yes, sir.
marker, tumor suppressor protein. And it has been
Yes, sir.
isolated, it has been like well established that if the concentration of this protein is, if it is goes down, then the tissue is likely to develop tumor. Okay. So
Yes, sir.
like in the process of development of a marker compound, first we need to isolate that marker and then we need to characterize it. Okay. So we have
Yes,
methods
sir.
of isolation and we have methods of characterization. Now the method of characterization is like you have, can you name some methods of you, what do you mean by characterization first?
So characterization means
Mm-hmm.
determining their structure,
Hmm.
their molecular pathways.
Hmm. No, characterization is something different. What is that? Hmm?
Sir, I don't know.
Now, characterization is you need to identify the molecule, and we have
Okay, sir.
mostly spectroscopic characterization of molecule. There are certain spectroscopic technique by which we can identify a molecule. Can you name any
Okay, sir.
spectroscopic technique which is used for identification?
Spectrum.
Can you name any spectroscopic technique?
Sir, mass spectroscopy.
Hmm? Yes, mass spectroscopy.
Yeah, yes, sir.
Anything else? So, there are different techniques of spectroscopy. There is a technique called UV visible spectroscopy, which gives information about the bonding like present in a particular organic compound, kind of bonding, whether it is say sigma bond, pi bond, or lone pair of electrons. Then we have IR spectroscopy, which gives idea about the functional group present in the molecule. Then we have mass spectroscopy, which gives us idea about the mass, and it is one of the chief technique for characterization of a compound. And then we have NMR spectroscopy, you might have heard about NMR spectroscopy, which gives idea about the like carbon-carbon or carbon-hydrogen bonding or carbon-nitrogen bonding. Okay, so it gives you idea about actually the position of atoms. So using all these four techniques, UV spectra, IR spectra, NMR spectra, and mass spectra, we identify the molecule, and the structure of the molecule, whatever you see in your books, is identified using these four spectroscopic techniques. Okay, so first we isolate a molecule, then we characterize a molecule, and after characterization, that molecule is established as a marker for a disease. Okay, so once the molecule is established as a marker, then no longer its characterization is required. So simply we do the quantitative estimation, and we can assign that whether the change in concentration of the molecule is responsible for the disease or not, and that molecule is developed as a marker compound. Like glucose is developed as a marker compound for diabetes, insulin is developed as a marker compound for diabetes, glycosylated hemoglobin is developed as a marker compound for diabetes, right? So these three marker compounds can be assessed to know whether the person is having diabetes or the person is normal. Okay, like for cancer, we have developed P53 protein as a marker for cancer. Okay, so these kind of marker molecule we develop, which are used for diagnosis. So for development of a marker compound, the primary requirement is that we should be able to isolate that compound, because unless and until, like if you see, if I give you a tissue, then the tissue is having millions of cells, and every cell is having a large number of biomolecules. Okay, so you have a huge, like very complex mixture of biomolecule, and if you want to analyze that complex mixture of biomolecule by spectroscopy, then it is not possible, because most of the spectroscopic technique demands that we should work with the pure compound, right? So you have to purify a compound. So purification of a compound is a great science, and if you are able to purify a compound, we can always use it for development of marker compound. So three techniques of purifications we have read. One is centrifugation, that I think I'll not be dealing with. Somebody else will be teaching that portion. And then chromatography and electrophoresis. So I'll be teaching these two techniques, chromatography and electrophoresis. So chromatography, as the name suggests, it came from a Greek word called chroma, and graphene means to write. So it is a separation technique, which is based on a concept of mobile phase and stationary phase. So I think before I go to the little bit history of that, I think I must tell you about one classical experiment done by Mikhail Swett. So what Mikhail Swett did actually, and the similar kind of experiment I have done a number of times in my experiment in my lab, and I have shown it to people. He took a cylindrical column like this, and he filled the column with the calcium carbonate. Calcium carbonate, you might knowing it is a just like it's a white powder, just like chalk powder, which we use as an adsorbent for painting of walls. Calcium carbonate, and that column was filled with calcium carbonate, and then it was percolated with a solvent called butanol. Okay, then what Mikhail Swett observed after some time, that he can see colored bands. So that's why the technique was named chromatography. Chroma means color, so bands were colored, and graphene means to write. It is something like you have a transparent column, and in that white column you have just drawn clear colored line with the help of a pen. So that's why the technique was called chromatography. So he was Mikhail Swett was the inventor. He was a Russian botanist, and he invented chromatography in 1901. and for during his research on plant pigments. Okay, so he separated chlorophylls, xanthophylls, and carotenoids using this technique. Okay, I'll share you this with you, this presentation. You can use it for studying so that you can write your exams at a later stage. Okay.
Okay, sir.
So these are the pigments he could separate, and this is one experiment which I conducted in my lab. You can see that a like this was the mixture was loaded here, and you can see a colored compound was separated. Okay, so since it was
Yes, sir.
used for separation of colored compound earlier, so the technique was called chromatography. But later on, we use this technique for separation of colorless compound. Like, can you tell me what is the color of protein? Yes.
So proteins appear colorless.
It appear colorless. Okay, then if white appear colorless.
Yeah, sir, white.
Yeah, why? Yeah, it is white, but white is white. So why some compound they are colored? So from where did you completed your MSc Summer? Your name is Summer. Summer is it? Eh?
Samra.
Samra. Okay, Samra. Yes, Samra.
Yes, sir.
From where did you did
Sir,
your
I've come.
MSc?
Sir, I've did my I've done my MSc from Kanpur, PPN College, Kanpur.
Okay, very good. Now tell me why some compounds are colored and why some compounds are colorless. Like why protein is
So
colorless?
certain compound, yeah, so because it absorbs every wavelength, colored wavelength. The compound that appears colors, for example, if certain compounds are green colored, so it means that they are absorbing every color except the green, and they are transmitting the green color. So so it appears green to our eyes.
Okay, good. So I'll just correct your definition a little bit.
Okay, sir.
Right. So when a compound is appearing colored, suppose something is appearing green. So that means it is only interacting with the green color of electromagnetic radiation, green color band of electromagnetic radiation. Rest it is not interacting with. Okay.
Okay, sir.
And now if a compound like protein is colorless, then that means it is not interacting at all with the colored region of electromagnetic.
Colored. Okay, sir.
You might be knowing what is the Lambda Max concept.
Hello, yes, sir.
What is the Lambda Max concept? Lambda Max.
Sir, can you please repeat? I cannot hear you properly.
What is Lambda Max?
Lambda X.
Yes. What is Lambda Max concept? You know, when a compound it interact with light, it absorb light under certain certain wavelength. Now the wavelength of radiation which is maximally absorbed by the compound, that radiation is called that wavelength is called lambda max. Okay.
Oh, okay, sir.
That radiation is called lambda max. Okay. So
okay, sir.
So the lambda max for if something is appearing green, then the lambda max falls in the green region. Okay.
Okay, sir.
If something appears colorless, that means the lambda max is in what? It is not in the colored region. Okay.
A colored
Not
region,
in
yes,
the
sir.
colored region. Okay. Now, can you tell me what is the wavelength of colored region of electromagnetic radiation?
Colored region.
Yeah. What is the wavelength?
Sir, it is from 300 to 700 nanometers something.
300 to 700. Huh?
Hello, sir?
It is no. It is not 300 to 700. You can say it is 400 nanometer to 700 nanometer. Okay. Or yes, you can say 400
sir.
nanometer to 800 nanometer. Okay.
Okay, sir. Okay, sir.
And below that, and what is the like, you know, the concept of Vibgyor?
Yes, sir.
What is that? You just tell me what is that? Yes, Samra. What is
Yes, sir.
Vibgyor concept?
Sir, Vibgyor's are the colors.
What is that? I want you to explain it.
Hello, hello. Yes, sir.
Yes, you explain it. I think you are walking.
Hello.
Are you walking?
No, no, sir.
Okay, I think by your it is becoming unstable very like
Yes,
frequently.
sir.
No? Your
Actually, sir. Yes, sir. Actually, I'm in my lab, and I think there's some signal problem here.
Okay, now I can hear you. Tell me.
Okay, sir.
Hmm.
Yes, sir. What were you asking?
What is Vibgyor? Full form of Vibgyor?
So, Vibgyor is the color combination of violet, indigo, blue, green, yellow, orange, and red.
But you have to expand it. Now, what is full form of it?
Sir, I don't understand you are saying what you are saying. Can you please
You
explain?
expand Vibgyor.
Hello.
You expand Vibgyor. It's a very simple question I am asking. Hello. Yes, Samra, can you hear me?
Yes, sir. Now I can hear you.
Yes. Now
There's
you
a glitch. I think that.
expand Vibgyor. Expand.
Sir, sorry, I don't know.
Okay, so it is V for violet,
Yeah, yes,
I
sir.
for
That.
indigo,
Yes, sir.
B for blue, G for green, Y for
Yes, sir. Y
yellow,
for
O for
yellow.
orange,
Yes, sir.
and R for red.
And R for red. Yes, sir.
Okay, so can you tell me
Okay.
that which color is having high energy, violet or red?
High energy.
Yes.
Sir.
So violet is having the highest energy and red is
Yes, sir.
having the lowest energy. Okay. And what will be the
Yes, sir.
wavelength?
Yes,
Which
sir.
one will be having the shortest and which one will be having the
Sir,
longest
reverse.
wavelength? Okay,
Sir,
reverse.
it's just the
Explain reverse.
opposite of the energy. Red has the longest
Okay.
frequency. Yes,
So
sir.
that means the
Yes, sir.
400 nanometer will be of violet and 800 nanometer will be of red.
Yes, sir.
Okay. So if a
Red.
compound is
Yes,
having
sir.
lambda max between 400 to 800 nanometers, it will appear what?
Yes, sir.
It will
Colored to
appear
our eyes.
colored. Okay.
Colored.
And if a
Yes, sir. Yes,
compound
sir.
is having lambda max below 400 nanometers, then it will appear colorless.
Colorless. Yes,
Okay.
sir.
Now, what is the lambda max of protein? What is the lambda max of protein?
Sir, I don't know.
Lambda max of protein is 280 nanometers.
Yeah, yes, sir. Yes, sir.
So if it is 280 nanometers, then it will be colorless. Okay.
Colorless.
So
Yes, sir.
initially, chromatography was used for separation of pure compounds, but later on we started even using it for preparation for the separation of colorless compounds. Okay. But still the
Okay, sir.
technique is called chromatography because it is used very frequently. Okay. So what
Okay,
I
sir.
do today, Samra, I am stopping here. I will
Okay,
share
sir.
this presentation with you. You go through the presentation and then I'll ask you so many questions. Okay.
Okay, sir.
Okay. And you have to read it so that you can understand it. Okay.
Okay, sir. Okay, sir.
So I'm stopping share. You want to ask anything from today's class? So
No,
when
sir.
we will start class tomorrow? So we'll start it at say around 9:30. because it need to be a 90-minute class. Okay.
So from 9:30, so can you please just do it from 10 o'clock?
Okay, 10 o'clock to 90 minutes means 11:30. Huh?
Yes, sir. Yes,
Okay,
sir.
so we can start at 10 and we can finish it at 11:30. Yeah? Because
11:30.
as per the
Okay, sir.
schedule, it is required that the class time should be of 90 minutes. But today we could only take a 1 hour class. But tomorrow onwards, it will be a 90-minute class. So sharp 10 we will
Okay, sir.
start and up to
Okay,
11:30 we
sir.
will go. Okay.
Okay, okay, sir.
Okay then. And
Okay,
I'll be
thank you, sir.
sharing this presentation with you. You go through it, read it. I'll
Okay,
ask you
sir.
many questions and then you just tell me
Okay, okay, sir.
what part you could not understand.
Okay, okay, sir.
Okay, okay, bye-bye.
Okay, okay, thank you, sir.
Mhm.